Okay, I am going to demonstrate your oxidation fermentation lab. Usually just call it the OF lab. And you've got some new features for this lab. So I'm going to run a demonstration. I'm just going to do like a partial. I'm just going to do one species. You're going to be using 6 different media tubes because you're going to do 2 different species. Each species gets 2 different treatments. So that's 2 tubes per species. And then you need 2 controls because, again, we have 2 treatments, the oxidation and the fermentation. So you're going to have 6 tubes total by the time you're done that are going to go into the incubator. And again, I'm just going to show you the inoculation part, so it's going to look a little different. I think you're going to be using NCECA and either E. coli or a different species called you can call it E. cloaca. Some people pronounce it E. cloaca, but it's going to look like word cloaca, actually named. We can talk about that later. I'm just going to do the NCECA today, I think. I'm just going to do the 1 species. So you're going to get an inoculated slant with your species. So I'm going to use my NCECA. And then you're going to get 6 sterile OF tubes, and these are a little different. They're still made of agar, but it's a special type of agar that has different ingredients from the nutrient agar you're used to working with. And one of the ingredients makes it very dark green. So this is your first biochemical metabolism test. And essentially you're seeing which species can oxidize, which one can ferment, and which ones can do both, which is why you need 2 media tubes per species plus your 2 controls. This will also be the first time you use more than 1 control. They're still both going to be negative controls because you are not going to inoculate them with bacteria. So they will be an example of a no reaction, which is what makes them negative as controls. So those are the best tubes, in my opinion, because you don't do anything to them. You're going to label them control. One's going to get the oxidation treatment and the other one's going to get the fermentation treatment, but other than that, that's it. You don't have to do anything further. The other 4 tubes you have to inoculate, and you inoculate them the same way, and then the difference comes out in the treatment. And basically for the fermentation tubes, you add a layer of mineral oil. So you're going to get a dropper bottle like this, and you're going to add about a centimeter or a milliliter of mineral oil to the top of the tube, and that's going to block out all the oxygen, which is what will encourage fermentation if the bacteria is capable. This lab is also a little different. You're used to working with slants, broths, and plates, and this one is different. You'll notice it's solid, but it's not a slant. It's got a flat surface. And for this, instead of using your loop, which you're used to using, you're going to use a needle. The idea is you use the needle to transfer so that you can go straight down the middle of your media. So I'm going to run you through how to inoculate these and then how to set them up for the 2 different treatments. Okay, first thing I'm going to do, actually before I set them down, is flame that needle. These do not get used a lot. In fact, I think you're only going to have 2, maybe 3 labs where you use these, including your unknown if you need this test. So I'm going to flame the whole thing. Sometimes I even bend it at an angle so I can get more of the loop into the flame. at once. Okay. So here's my inoculated culture of N-Zika. And I've got a little bit of concentration up at the top here. So I'm going to cool the needle on the side. I'm going to pick up the N-Zika. I'm just sort of swishing it around. And I'm zigzagging the needle back and forth through the culture to get my transfer. And then uncap. Put that down so you can see what I do. I'm going to go right down the middle. And the challenge of this inoculation method is I want to go straight down. I don't want to touch the bottom. And then I want to pull it out exactly where I put it in. So you don't want to jiggle your hand too much. You need kind of a steady hand. I'm going to flame the top of the tube before I put my cap on. And then this one's ready to go because I labeled this one as not getting the oil. Basically, I just didn't write anything besides the N-Zika. And then the letters OF are on there because, again, this is the OF lab, Oxidation-Fermentation. I'm going to flame my loop again really quickly. And then I'm going to do the exact same thing for my fermentation tube. Because, again, the difference is going to come out with the oil. So, again, I'm just going back and forth, trying to pick up the N-Zika with the needle, not the agar, so I'm scraping it across the surface. I'm not digging in to the agar. You can typically see it when you get the inoculum on the needle. And then I'm going to uncap the tube, and then I'm going to go straight down. Don't want to touch the bottom, but I want to go down as far as I can, maybe about 1 centimeter, 2 centimeters away from the bottom. Pull it straight back out. Flame it, cap it, and then this one's labeled oil. So I'm just capping it for a second while I flame my needle so that I can lay it down in good conscience. And then when I'm ready for the oil, I'm going to uncap it again. I'm going to open my dropper bottle of mineral oil, and then I'm going to pour the oil down the side of the tube. So that it kind of builds up on the top. Slowly, carefully. And again, I want about, 1 centimeter or so of oil. I just want it on there enough to block where I pierced the media, because I don't want any oxygen to get through here. And then these are ready to go. So you're going to do that exact process again for your either your E. coli or your E. cloaca, whichever one I end up going with. And then you're going to set up your two controls, omitting the inoculation part. You're just going to go ahead and open your oil, pour in the oil, and then cap it. Your control for oxidation, you won't even open. Just label it control with no oil, and it's ready to go. So you're going to have 6 tubes total. You're going to put those in the incubator, and when they come out, the way we decide who is able to digest the carbohydrates in the media, and I think I'm going to use glucose for you, is it will turn yellow. So here's 2 examples of some tubes that have had some oxidation, so you can see the difference. This is a slow oxidizer, so it's not totally yellow. It's kind of greenish yellow, but you can see when you compare it to a tube that hasn't gone through the metabolism, you go from really dark green to a much lighter color, and typically it goes all the way to yellow. So the idea is, if in your oxidation tube After incubation, if it turns yellow, that means that this is a bacteria that's capable of metabolizing glucose in the presence of oxygen. If you have yellow in your oil tube where there was no oxygen, that tells you you had a bacteria that's capable of fermentation. So it's able to metabolize the glucose using that metabolic pathway. And remember, fermentation is an alternative to cellular respiration. If there's no oxidation after the glycolysis, the cell will go into fermentation. Keep it straight and separate from anaerobic respiration, which happens at the end of respiration. So anaerobic respiration is not an alternative pathway to respiration. It's a part of respiration. Fermentation is an alternative pathway. Once you get those in the incubator, you're done. You're free to go. So you won't be able to finish this lab until you read results the next time. And I'll see you